Hello, everyone. My name is Amber, and I'm a PhD student at the Laboratory for Genetics of Malignant Disorders at KU Leuven. And today I will present to you a part of my PhD project, which is on how optical genome mapping detects pathogenic aberrations in non-Hodgkin lymphoma and reveals additional information. So first, I just want to show you how we use optical genome mapping here in Leuven. So as you can see, we use it on a lot of different hematological malignancies. A few years ago, we started with acute leukemias, but now we've expanded this to a lot of different other types of hematological malignancies. And of course, we're also part of some consortia and user groups to share and compare optical genome mapping results with other people using it on the same diseases. Okay, so let's start with what non-Hodgkin lymphomas are. So this is a heterogeneous hematologic malignancy, and it arises from the clonal proliferation of mostly mature B, T, or NK lymphocytes. It's heterogeneous, meaning that there's over 60 different entities recognized by our classification systems. And this classification is mostly based on the morphology and immunophenotype of the cells, but also on clinical features and genomic information. So most of these entities are characterized by structural variants and copy number alterations, which can give us a diagnostic profile with biomarkers. And integrating this information into the classification system has improved diagnostics, but some subtypes still have a paucity of biomarkers. This is mostly the case in the rare subtypes where we have little genomic information. So currently, the cytogenomic analysis of non-Hodgkin lymphoma mostly consists of two technologies. The first one is chromosome bending analysis, or CBA. And this is a visual evaluation of structural variants and copy number alterations, but it has a quite low resolution of around 5 to 10 megabases, and it can be even lower in case of low quality metaphases. And in case of complex results, sometimes aberrations can be overlooked. And one of its main drawbacks is that it requires fresh tissue for cell culture, and that's one of the main reasons why technical failure or uninformative results can occur. And this can be either due to the fact that the sample was too small to start a cell culture, or it could be degraded during transport, but there can be other reasons as well. Then there's the second technology often used, which is fluorescent in situ hybridization, or FISH. And this has a better resolution of around 200 kb, but it's a targeted approach, so you need a priori knowledge of which loci need to be tested. So that means that anything that is new or unexpected will not be detected. There are some other technologies that can be used, such as NGS panels, low-pass sequencing, or different kind of arrays, but all of these come with their own limitations. So that's why we went looking for a technology that can detect both structural variants and copy number alterations in one single assay without needing a cell culture. And that's where optical genome mapping comes in. So as you know, this is a DNA-based technology that gives us a genome-wide view of structural variants and copy number alterations. Because of its high sensitivity, we get an increased detection of pathogenic variants, but we can also detect novel variants that we didn't know were present before. And because it is DNA-based, it also allows us to rescue cases where CBA fails. So to determine whether optical genome mapping detects the pathogenic aberrations in non-Hodgkin lymphoma that we wanted to detect, we first have to define what these aberrations are. And we did that with a tier-based system. So we have our tier 1 characteristic aberrations. These are disease-defining for a certain subtype. One good example for this is MYC rearrangements in Burkitt lymphoma. We also have the tier 1 other aberrations. These are strongly associated with a certain subtypes, but they are less specific. And one good example of this is the gain of 9p in primary mediastinal B-cell lymphoma. And then lastly, there's the tier 2 aberrations. So these are recurrent in a particular NHL entity, but they can also occur in other entities, meaning they don't really help us to subclassify a certain case. And one good example of this is the gain of chromosome 3 in many different non-Hodgkin lymphoma entities. So we recently published this paper on optical genome mapping in non-Hodgkin lymphoma, and here I will show you our main findings. So we included 110 different samples, and as I mentioned in my introduction, non-Hodgkin lymphomas are very heterogeneous, so we tried to include as many different subtypes as possible, which is what you can see in these pie plots here. So we included 79 B-cell lymphomas and 31 T or NK cell lymphomas. As non-Hodgkin lymphoma is a lymphoid disorder, most of the cases will arise in lymph nodes, which is what we also saw in our cohort, but we managed to include some other types of samples, which is what you can see here. And as a proof of concept, we also tried to include a few different sample types to not only rely on fresh frozen material, and those are these. So cells suspended in EDTA or cell pellets, and these types of samples also work. So in total, we got informative OGM results in 94% of our cases, but we did see that 7 cases failed OGM. In 4 of these, that was due to too little starting material, and the remaining 3 just had bad DNA quality. And we don't really know why this was the case. So our main finding is that optical genome mapping is highly concordant with the standard of care, and it can also detect additional biologically relevant information. When we look at the tier 1 characteristic aberrations, which is what you can see here, we see that optical genome mapping detected 54 out of 55 tier 1 characteristic aberrations, while the standard of care only detected 51. So optical genome mapping did miss 1 rearrangement. This was a MYC rearrangement in a large B-cell lymphoma, but this rearrangement was present at a very low allele frequency, meaning it was right at the border of the limit of detection of OGM. And as you can see, there was also 4 extra aberrations detected with optical genome mapping that were not detected by the standard of care. When we look at the total number of tier 1 and tier 2 aberrations that were reported by the standard of care, optical genome mapping detected 168 of them. And again, we did miss some aberrations, 13 to be particular, but these were all again present at the border, at the limit of detection. So in total, we saw that there was a concordance of 93%, and that the limit of detection, similar to what other people have seen, is around 5 to 7.5% of variant allele frequency. And then, as you can see here, optical genome mapping also detected 77 additional aberrations. About half of these were above the resolution of CBA, but they were missed either due to the quality of the metaphases or because they couldn't be recognized. And about half of them are below the resolution of CBA, so they could never be seen by this technology, but they can be seen by optical genome mapping. So we also included 32 cases with CBA failure, and in 27 of these, optical genome mapping was able to successfully rescue these, and we did have informative results. But 5 cases also failed OGM, so they had a failure of both CBA and OGM. And we think this is probably due to sample-related issues rather than a technology-related issue, but we're not 100% sure. Okay, so now I just want to show you a couple of examples. So first we have this follicular lymphoma case. So here you can see the CBA results. So there is additional material on 6q. We have a translocation between both P arms of chromosomes 8, and then the typical translocation for follicular lymphoma between 14q and 18q, as well as a derivative chromosome with the same translocation. And then here you can see the circus plots with the optical genome mapping results. So on the outside, there are the cytobands. Then we have the structural variant track, the copy number alteration track, and in the middle, there are the translocations. And we see that optical genome mapping detects the same aberrations that were reported by CBA. So we see the 14-18 translocation that juxtaposes IGH and BCL2. But we also see that there is a rearrangement here at the BCL6 locus that we didn't know was there before. So here the translocation of BCL6 goes to chromosome 6q, and we also have a gain telomeric of this region. So we can assume that this additional material on 6q that we saw before was actually this rearrangement with BCL6. And BCL6 rearrangements are recurrent in follicular lymphoma, mostly in the more aggressive type, so the grades 3A and B. But we wanted to be sure that this aberration was a real aberration, so we confirmed it with FISH. And that's what you can see here. So we use the rearrangement probe with a green centromeric signal and a red telomeric signal, and BCL6 right here in the middle. And then we detected two clones. So first there was a balanced clone with one normal colocalized signal that has a green and red signal together. And then there's one separate green signal, one separate red signal, meaning that there's a rearrangement. Then we have our second clone here. Again, you can see one normal colocalized signal, a separate green signal, and two separate red signals, which means that there is a gain of this telomeric, of this region telomeric of BCL6, which is what we saw in our optical genome mapping results as well. So here there is another example case. So this is a diffuse large B-cell lymphoma, where we did not have any CBA results. We did have FISH, which is what you can see here. So there were 3 balanced rearrangements in BCL6, in MYC, and in BCL2, making this a triple hit lymphoma. And here you can see the circle spot of the optical genome mapping results. So again, we see the same rearrangements that we saw with FISH, but we get additional information from optical genome mapping. For one, we get the partner chromosome for every rearrangement. So in the case of BCL2, we now know that it's a rearrangement between IgH and BCL2, for MYC with MYC and IgL, and BCL6 with chromosome 5q. Not only do we get our partner chromosomes, we also get additional aberrations, such as a gain of 12 and deletion in 9p, both recurrent aberrations in diffuse large B-cell lymphoma that we didn't know were there before we did optical genome mapping. Okay, so now just to conclude all of this, first, we know that optical genome mapping is very concordant with CBA and FISH, with over 90% of the aberrations reported by standard of care also being detected by optical genome mapping. Secondly, optical genome mapping reveals additional biologically relevant information, for example, by detecting additional aberrations. And then lastly, optical genome mapping can rescue CBA failure by giving us informative results where there were none before. Okay, then all that's left for me is to thank you for your attention. And I'd also like to thank my supervisors as well as the lab at the hospital and my collaborators.